My name is Tara Martinez, and this summer I have the honor and privilege of working under the mentorship of Dr. Mark I. Green with the assistance of Dr. Gail Massey, Dr. Hung Tao Zhang, and Dr. Gao Ping Dong in the Department of Pathology and Laboratory Medicine at the University of Pennsylvania on a project entitled Expression of GST PIM2 Fusion Protein. Before I get into the specifics of my research, I'd like to give you some background information. As Francis Crick once said, DNA makes RNA, RNA makes proteins, and proteins make us. The first step to a protein synthesis is the replication of DNA. In order for DNA to replicate, both strands of the DNA must denature and separate. Then primers and polymerase are added to the strands of DNA to bind and replicate to a second strand of DNA. Once the DNA has replicated, a portion of that DNA is transcribed into mRNA, and using tRNA, ribosomes, and RNA, mRNA translates into proteins. DNA, also known as deoxyribonucleic acid, is found in the nucleus of all cells. It holds genetic information, and it controls the development and functioning of all living organisms. RNA, ribonucleic acid, is very similar to DNA, but one of the main differences is that RNA is not missing an oxygen group in such DNA is, hence why it's called deoxyribonucleic acid. RNA catalyzes biological reactions, it controls gene expressions, and is one of the main contributors to a protein synthesis. Proteins are made up of 20 amino acids, and they are found in all of our cells. They catalyze biochemical reactions, they control cell signaling, as well as structural and mechanical functioning, as well as many other things. Fusion proteins is when you take two different proteins and you fuse them together so that you make one protein. The reason why you did this is because you'd like to express both proteins as one protein. And once you express them as one protein, you can use it to purify that protein. In our lab, we use plasmid vectors. And a plasmid vector is a portion of the host bacteria that allows the DNA, or gene that we're looking at, to replicate. Shown in the picture on top is a host bacteria replicating by itself. But here on the bottom shows plasmid entering the vector, and then the host bacteria replicating and letting the vector replicate in both host bacteria. A kinase is an enzyme that modifies other proteins through a process called phosphorylation. Phosphorylation is when you add a phosphate group to a protein to change its function and this can change the function of its enzyme substrate. In our lab, we use PIM2, and PIM2 is a serine-threonine kinase. It is found in most cells because it controls the growth and metabolism of the cell, also controlling whether or not a cell apoptosis. To the right shows a crystallized form of the protein PIM2. Now, GST is a family of enzymes. We use it in a fusion protein to purify the other protein. And in our case, we're using it to solubilize PIM2. Again, to the right is a picture of the crystallized form of the protein GST. This leads me to my research design. The purpose of our experiment is to express and purify PIM2 in E. coli cells. And our hypothesis is if PIM2 is cloned in a PGX 5x3 vector, it will be expressed as a GST fusion protein. Here I list my materials. Please note that the most important are highlighted in green. Please take a second to look them over. This leads me into my methodology. First, we did a PCR to replicate the DNA. And then we did a digestion to cut the vector in which the DNA will be inserted. From that, we did a ligation. And using the ligation product, we did a transformation and a mini-prep. After we removed the DNA from the mini-prep, we sent it to screening and sequencing. And then we did a protein expression and a protein purification. PCR, also known as polymerase chain reaction, is the first step to a protein synthesis. It replicates the DNA in 3 different stages. First, it denatures the DNA and separates both strands of DNA. Part of annealing, it adds primers, polymerase, and DNTPs to bind to the DNA strands. Then in extension, a Taq protein is added to each side in reverse directions to to the primers and start extending on the DNA replicating. This step must be done 30 to 40 times so that the exact portion of the DNA that we want replicated does in fact get replicated, and not any other portion of the DNA. During our digestion, we cut our plasmid vectors using restriction enzymes. In our lab, we use the restriction enzymes EcoR1 and BAMH1. After the vector is cut, we run it on a DNA gel to make sure it is the proper size to insert our DNA. In a ligation, we do exactly that. We take our complementary DNA and we insert it into our vector. During a transformation, we heat shock the competent cells with the ligation product. Please note that the competent cells used in a transformation are different from the competent cells used in a protein expression, which you'll see later. In the transformation, we use a DH5 alpha competent cells. After the competent cells have been heat shocked with the ligation product, we incubate it in an LB medium at 37 degrees Celsius. Then we plate and culture the cells overnight. After the cells are cultured, we take colonies from the cells and we elute the DNA. First, we resuspend the cells, and then we add a base buffer and a neutralization buffer. The base buffer lyses the cells, and the neutralization buffer neutralizes the base buffer. After that, we wash out the cells for any other impurities, and we elute the DNA. Once the DNA is eluted, we send it to screening and sequencing to make sure that the proper DNA has been cloned. If it becomes successful, we move on to a protein expression. In a protein expression, we're using our DLX competent cells, and we culture it at 37 degrees Celsius, and then we induce it at 16 degrees Celsius in a 0.5 millimolar IPTG. An IPTG is just a chemical used to induce the expression. After that, we harvest and lysate the cells. Once the cells are lysated, we move on to a protein purification. Shown here is a picture of the steps to a protein purification. First, we add the glutamine mag beads and then incubate for 30 minutes to an hour. While the glutamine mag beads are incubating, they attach to our selected protein. After they're incubated, we wash out the solution of any other impurities and take out our target protein. This leads me to my results. Here is an overview look at the 4 DNA and protein gels that we ran. First, we ran a DNA gel after PCR to make sure that we had the correct DNA cut. And this shows PIM2, the DNA that we wanted cut at the exact size we wanted, 1,000 kbs. Then we ran another DNA gel, just to show the different sizes in the vector that we're trying to clone PIM2 in, and exactly where PIM2 lands. Then we took 4 clones of PIM2 that we cultured, and we induced them into our vector. Shown here is that all 4 clones successfully were induced in our vector. If they weren't induced in our vector, they would have ran through and would have landed where PIM2 normally is, and this would have been highlighted. And lastly, we ran a protein gel. And the first column shows GST by itself, and then the second column shows GST when added to PIM2 that was cloned in our PGX5X3 vector. This shows whether or not PIM2 was cloned as a GST fusion protein. This leads me to my discussion. Before I conclude, I'd like to reiterate my hypothesis, which is that if PIM2 is cloned in a PGX5X3 vector, it will be expressed as a GST fusion protein. And this hypothesis was accepted, because as shown in the picture before, PIM2 and GST, when fused, did fuse as a fusion protein. Due to the fact that PIM2 fused as a GST fusion protein, we can move on with our research in a in vitro kinase assay. This allows us to study the function of the protein we just made and study its correlation to TRIGs, which are regulatory T cells. Here are my acknowledgements.